Welcome back everyone to the Swalife podcast series. Today, we are diving deep into a term that sounds technical but actually decides the fate of most drug discovery programs. That is lead identification. Dr. Praveen Madhe, this is one of the those stages people underestimate. Is it right?
Absolutely. Most people think that the drug discovery fail in clinical trials. But when we look at the reality, basically many failures are already at the phase of lead identification. If you start wrong, everything which is downstream just going to magnifying that mistake. So basically, initial stages of the drug discovery where target and lead identification is done is the important stage.
Right. So let's start from the basic now. What does actually lead identification mean?
Okay, so when we talk about lead identification, it is a step where we decide which molecules deserve our trust, time, and money. So you have already identified the disease, the biological mechanism, and one or more targets. And now you are working on lead identification. So it is important to understand which molecules can realistically modulate this biology in a way that could help patients. So that's what we need to ask. Is critical because every later step, that is optimization, toxicity, trials, depend on this choice. A weak lead creates false hopes and actually it wastes a lot of years. And a strong lead can actually survive multiple scientific setbacks. So it is a critical step, thinking of it as a choosing the foundation of a building. No matter how beautiful the upper floors are, a bad foundation is going to collapse the building. So it is very important.
That's very rightly said. Also, there is always confusion about hits, leads, and drug candidates. Can you break this down simply?
Yeah, sure. This is actually, very important to understand because a lot of misunderstanding is happening here. When we think about heat, it is simply activity. A molecule does something in an assay, so that is a heat. Where a lead is a trust, the molecule works and it show the reproductively result. Then it shows selectivity and actually does not or have less toxicity. So that is the lead, and a drug candidate, which is from the lead, which is a commitment. That is, you are ready to invest serious resources. So hit initially just give us that it might actually have something. In the lead, we actually perform different assays, and it shows us the similar results, and based on that, that lead goes into the drug candidate. When it goes into the drug candidate, we can actually take it further into clinical phases and in the formulation. So a lead must answer three silent questions. Does it make biological sense? Can it be optimized? And is it realistic for humans, not just for cells or for the animal? So most hits never earn that trust.
I hope this answer clears the classic misunderstanding between the three terms. Let's move ahead. So, how does lead identification? How was lead identification done previously, traditionally?
So in the traditional way, wet lab was the the dominance part in the lead identification. So like doing high throughput screening, where thousands of molecules or the compounds were screened. Then also the second was the natural product isolation and repeated testing of those natural product. Single target biochemical assays were utilized, And when we look at these traditional methods of lead identification, it is actually massive cost. We have to pay for that. Then long timelines are taken, and the success rate is very low. So there is a poor understanding of why a molecule works. And mostly, in the traditional approach, the complexity of the disease is ignored. So human disease are network, not switches. So that is very important, which need to be understand.
Yes, and as we move from traditional approaches to modern approaches basically and mainly artificial intelligence. So, where does artificial intelligence change this game of lead identification?
So in, like, previously the leads, we wet lab was the dominance, but then there was in silico studies where computer based studies, tools, the databases were used. But now, including AI actually has changed the order of thinking. So it's not that the AI just started into the drug discovery, it was there, but the order of thinking has been changed. So instead of having compound essay and then hoping for result, now with the AI, you are actually looking at the disease biology, you are looking at the pathway networks, you are looking the target confidence, and then based on that, you are prioritizing your molecules. So this artificial intelligence has allowed us to predict the interaction before the experiments. Then it is also integrating genomic, proteomic, then the literatures, then the real world data. So that is again something is useful. And based on that, focus lab work only can be done. So that way, it is going to eliminate the problems which can occur in wet lab. But yes, it is not going to remove the wet lab from it, because it is going to help to protect them from bad bits. So we will still have the wet labs, but the wet labs are not now focusing on non-relevant molecules, but it is going to be more focused molecules.
Yes. So, what biological data is actually important when we have to choose the leads?
Okay, so that's actually a great question here. So there are three layers which actually matters. So the first is that the target validity. So genetic evidences are there, expression in disease tissue is there, and there is a causal, not just correlated roles are there. So target validity is there. Then the second layer is the pathway context. Is the target central or peripheral? Does it modulating it restore balance or cause just the chaos? Then is it disease relevant? Does the human data always overweight animal only data? Are there clinical correlation available which actually matters more than the isolated experiment? So when we look at the good leads, good lead identification is about biological confidence, not just essay performance. So the result should be more biological confidence.
Yes. In the previous episodes, we have been talking about the Swalife AI Discovery and Study Design Studio. So, as we go deep, how does this studio actually connect to lead identification and what difference does it make?
Before actually starting with the AI or developing the Swalife AI Studio, uh, we were using network pharmacology and also in silico models. But then we realized that the data which is already there in public domain, people are already utilizing animals and for human studies, but still the leads that are the molecules are not coming in the market. So that was where we started with Swalife AI Discovery Studio. So when we look at most platform, they are predicting the target or they're doing the docking, the in silico study. But with our studio, we actually design as a decision making engine. So it is actually helping us as an assistant. It is not just prediction. tool. So if we look the step by step, the step one, we begin with disease phenotype, clinical pattern, known failures and limitations. So this prevent actually chasing some irrelevant targets. So that's the first step. In the second, instead of one target, we analyze the pathway, the crosstalks. Then, are there any redundancy or compensation between the targets? So, network level target intelligence we done. This is a critical for some of the chronic diseases such as cancer, then inflammatory diseases and neuro degenerative diseases. And in the third step, what we do multi-scoring lead evaluation. So each potential lead is evaluated for biological relevance, then safety, then translational feasibility. This is where AI becomes strategic, not just the computational. So we are actually getting information about that at multiple scoring. In our case, we are utilizing it for herbal and multi-target advantages. So that is helping us with step four, where classical drug discovery actually struggles with herbal and combination therapy. In our system, we have built multi-compound logics, then synergetic mechanism, and connecting it with the network pharmacology. That's why it works so well with our precision herbal and integrative therapy that we actually now moving from preclinical to clinical work. So in step five, it helps us study designing integration. So the lead which we have identified does not end with just the molecule. It actually give us information that what type of in vitro assays, in vivo assays, and then how to move towards the clinical observational design. So this lead is born with the development plan. It is not just having activity data.
So, with the tool, you are not just identifying needs, you are pre validating their future. Is that so?
Yeah, exactly. We don't just ask, does this molecule work? We actually ask that, does this molecule deserve a future? Because as I already mentioned, the lead need to be moved into the further clinical studies.
Yes. So, the next question I'm going to ask is especially important for the students who are listening. If someone wants to work in need identification today, what skills really matter?
This is crucial because lead identification role now have changed dramatically. When we look at the old skill, it was old setup of the lead identification validation. Single essay expertise were in the demand. Then having narrow lab specialization was important. Then trial and error mindset. Okay, I am performing, I'm having issue, I will perform again. So that mindset was there. But now with the modern and with the addition of the AI, lead identification scientists play important role. Not only about working on the lead, but they have to look at the disease biology. So they will be disease biology literacy, where they will understand that how the mechanism work, not just memorizing the pathways, but how this mechanism is working. That is one. Second is data interpretation skill. So knowing how to judge evidence quality is very important. So that is something the student need to develop. Then we're talking about AI. So AI and computational fluency. So not just the coding expert, but also intelligent usage of those AI tools. You need to gain those skills. Then systematic thinking, that is seeing interaction, feedback loops, and trade-off is important as a lead identification scientist. And transnational mindset, where always asking, will this survive in the human? Because a lot of time, a lot of discoveries in cells, in animals, even in some of the primary clinical studies shows it promising. But then what happens about that lead? Sometime it don't move into the market, and there are different reasons. So the career path, student can grow into roles such as AI assisted discovery scientist, then analyst, network pharmacology or data analyst, then translational research strategist is another role, then computational experimental scientist is another, which actually help to bridge between in vitro or the initially clinical, even the preclinical and clinical studies. So at Swalife, we actually have different project, and we have created this project-based program where the students or even the faculties who are working in the academic can be part of it and work on real lead identification pipelines and using the tools which we have developed, which will help in decision making and be part of the publication, the products, and even the patents. So actually, we created industry-ready professionals, not by training them, but being part of this program-based, project-based program. So you are not just going to get the certificate, but you actually working on the, in the drug discovery programs.
So, does that mean that lead identification is not just a lab role anymore?
No, it's not at all now. It's becoming one of the strategic, high impact role in drug discovery.
So, before we pack up, what's one takeaway you want listeners to remember from this episode?
Lead identification is not about finding active molecule. It is about finding worthy starting point. This is very important. AI, when using responsibly, does not replace the scientist. It actually help in amplifying the judgment. And the future actually belongs to those who are combining the biology, the data, and the strategy. So that's from my side.
That was a very powerful conclusion. Thank you, Dr. Praveen Madi, and thank you to everyone listening. We will see you in the next episode. Thank you.
Thank you.